For the longest time, we've treated many cancers as if they're one single disease. But what if that was just fundamentally wrong? A really groundbreaking discovery is now revealing that one of the most aggressive childhood cancers isn't one disease at all, but two, and they've been hiding in plain sight. And honestly, this changes everything.
This is the question that keeps oncologists and researchers up at night. You can have two patients, same age, same exact diagnosis, getting the same treatment, but one responds perfectly while the other one struggles. It's a dangerous and frustrating mystery, and today we're going to follow the clues that are finally starting to crack this case wide open.
So let's zoom in on our case file for today. We're talking about Burkitt lymphoma. It's pretty notorious. It's fast-growing, incredibly aggressive, and it's actually the most common type of non-Hodgkin lymphoma in children. It's a really tough opponent, and for a long time, its behavior has just been, well, a total puzzle.
So, what exactly are we dealing with here? At its very core, Burkitt lymphoma is a cancer of our B cells. Those are the immune cells responsible for making antibodies. Its genetic signature, the thing that makes it tick, is a very specific mutation that basically jams the accelerator on a gene called MYC. This forces the cells to just divide and divide, completely out of control.
For years, doctors had a sort of checklist of clues to try and figure out how this disease would behave. They'd look at where in the world the patient was from, what their age was, and whether their tumor was linked to the Epstein-Barr virus. But here's the kicker. These clues just weren't enough. They couldn't solve that central mystery of why the outcomes were so wildly different. The story was missing a crucial chapter.
So to find that missing chapter, researchers knew they had to look somewhere else, somewhere beyond the DNA sequence itself. They needed to investigate a hidden layer of information, one that controls how our genes actually get used.
And that hidden layer? It's called epigenetics. Now, I know that word sounds super complicated, but the analogy is actually really simple and kind of brilliant. Just imagine your DNA is this massive instruction manual for how to build and run your cells. Epigenetics are like the sticky notes and highlighter marks that get put on the pages. They don't change the words in the book. The genes are still there, but they tell the cell which instructions to read and which ones to skip, effectively turning genes on or off.
And this is where it gets really crucial for understanding cancer. In a cancer cell, this whole system gets hijacked. The cell starts slapping these epigenetic sticky notes all over the wrong pages, silencing the very genes, the tumor suppressors, that are designed to slam on the brakes and keep growth in check. This epigenetic chaos basically gives cancer a massive, unfair advantage.
So, armed with this idea, a team of scientists analyzed the epigenetic patterns from 218 Burkitt lymphoma patients. They were basically hunting for a pattern in all these sticky notes that could finally explain the different patient outcomes. And what they found, well, it changed everything.
So what was it? When they dug through all of that data, did a clear pattern actually pop out from all that noise?
Oh, it absolutely did. The data cleanly split the patients into two totally distinct groups, which they called epitypes. First, you've got HypoBL. Hypo just means less. So these tumors had fewer of those gene-silencing sticky notes. And then you have HyperBL. Hyper, of course, means more, and these tumors were just covered in them, silencing a whole bunch of important genes.
Now, this is really interesting because it connects back to one of those old clues, the Epstein-Barr virus. You can see right away that HyperBL is a bit more likely to be virus positive. But, and this is so important, you find both epitypes in patients with and without the virus. This was the proof that this epigenetic signature was a fundamental thing, not just some side effect of a viral infection.
Okay, so now we have our two suspects, HypoBL and HyperBL. And when we start comparing their criminal profiles, so to speak, we see that their differences go way deeper than just those epigenetic sticky notes.
When you put them side by side like this, it's pretty clear they almost look like two totally different diseases. HyperBL has way more genetic mutations. They're each driven by different key mistakes in their DNA. And what's really fascinating is that they seem to start in different kinds of cells. HypoBL looks like it comes from a more mature memory B cell, while HyperBL comes from a younger B cell that's still being trained in the immune system's boot camp, a place called the germinal center.
And wow, this chart really drives that point home. Just look at the bar for HyperBL. This is not a small difference. These tumors are genetically way more unstable, way more chaotic. They're just riddled with mutations compared to the HypoBL tumors.
This whole cell of origin idea, it's a huge piece of the puzzle. It suggests these aren't just two different flavors of the same cancer. They might actually be two different cancers that start in completely different types of B cells at different points in their lives, which then just happen to end up looking similar under a microscope.
But you know, as fascinating as all that biology is, the discovery that truly matters, the one with real-world human impact, it had nothing to do with the cell's past. It had everything to do with the patient's future.
And here it is. For adult patients, the difference was just stark. You can see it clear as day on the chart. Patients with the HyperBL epitype had significantly worse survival rates. That p-value you see, the 0.014, that's just the statistical way of saying this result is absolutely real and highly unlikely to be a fluke. This wasn't just some biological curiosity, this was a life or death predictor.
So there you have it. By digging into this hidden epigenetic layer, the researchers finally found the clue they'd been searching for all this time. They cracked the case of the unpredictable outcomes.
And listen to how the researchers themselves put it. They found that this new epitype classification was a much stronger predictor of the cancer's behavior than any of the old clues, like whether the virus was there or not. It's a whole new layer to understanding this disease.
So let's bring it all back to what this really means for patients. It means this discovery could soon move out of the lab and into the clinic. It could give doctors an incredibly powerful new tool. Just imagine, right from day one, they could analyze a tumor and know if they're dealing with a HypoBL case or a much higher risk HyperBL case. And they could tailor the treatment plan based on that knowledge.
And that leaves us with one last really exciting question for the future. If these epigenetic sticky notes are causing the problem, can we develop drugs that basically act like an eraser? Could future treatments be designed to literally wipe these marks clean, reawakening the very genes that are supposed to stop cancer in its tracks? Now that might just be the next chapter in this incredible story.